When we talk about the very beginning of a new life, there is this almost universal expectation of a single magical flash.
Right, like a flipped switch.
Exactly. A tiny spark in the dark and boom, a new organism exists.
Mhm.
It's a very comforting cinematic idea. But, I mean, that's not the whole story, is it?
Not even close, actually. When you actually look at the biology, when you track the microscopic reality of how a haploid sperm and a haploid egg fuse, that cinematic instant just doesn't exist.
Right.
We are looking at a biological landscape that is precarious, deeply complex, and, unfolds over a surprisingly long time.
Yeah, it is not a moment at all. It is a roughly 24-hour biochemical cascade. So
Okay,
let's unpack this.
let's do it.
If you, the listener, are picturing fertilization as a quick spark,
Yeah.
I want you to just erase that. Instead, imagine it as a wildly complex computer system booting up.
That's a great analogy.
Thanks. It's a system where a dozen different passwords have to be entered and physical keys have to be turned in an exact, non-negotiable sequence over a full day.
And if any single one of those microscopic keys fails to turn perfectly, the entire system just shuts down. The sequence simply terminates right there.
Wow. So that is our mission for this deep dive. We're going to track the incredible, perilous journey of how a new organism actually begins, from the first encounter between cells to the formation of a true embryo.
It's quite a journey.
It really is. And whether you are endlessly curious about human biology or maybe you were trying to understand the science behind modern fertility medicine, this journey is full of aha moments that will, forever change how you view your own origin story.
Absolutely. And to truly appreciate just how complex this 24-hour boot-up sequence is, we first have to look at how wildly wrong humanity was about it for centuries.
Well, the history here is wild.
Yeah, it really is. The evolution of our understanding wasn't just a matter of changing ideas. It was a matter of slowly inventing the physical tools needed to actually see the truth.
Right, because for the longest time, two rival theories completely dominated the conversation.
Right, epigenesis and preformationism.
Yeah. One was epigenesis, tracing back to Aristotle and then revived in the 1600s by William Harvey.
Right.
And that basically said an embryo's structures arise gradually out of unstructured material.
Which is closer to the truth. Yeah.
But it competed heavily with preformationism. And this was the idea that a fully formed miniature organism already existed and it simply, grew larger, like a sponge expanding in water.
And they took this incredibly literally. In 1694, a scientist named Nicholas Hartsoker sketched what he called a homunculus.
A homunculus.
Yeah, he drew a fully formed, tiny human being just curled up in the fetal position inside the head of a single sperm cell.
Wait, I have to push back on this. So, for over a century, brilliant scientists literally believed we were just miniature humans waiting to inflate like a balloon.
It really did.
How did they possibly explain the mother's genetic traits? If the whole human is just riding in the sperm, where do the mother's eyes or hair come from?
That is the exact logical wall they kept hitting. The problem wasn't a lack of intelligence. They simply couldn't see what was happening on a cellular level. Because they didn't have the microscopes for it yet.
Exactly. The slow march to the truth required painstaking physical experiments to bridge that gap.
Yeah.
For instance, in the 1780s, an Italian scientist named Lazzaro Spallanzani did something brilliant with frogs.
Okay, what did he do?
He physically filtered frog and toad semen. By separating the fluid from the physical cells, he proved that the actual microscopic particles, the sperm themselves, were required for fertilization.
So if you just use the filtered fluid, nothing happened.
Exactly. Nothing happened. You needed the physical cell, not some mystical vapor or aura, which was, the prevailing belief at the time.
An aura. Wow. So how did we finally get to a place where we could actually see both sides of the equation?
Well, it took decades of advancing optics. In 1827, Carl Ernst von Baer finally identified the mammalian egg itself.
Which was the big deal.
A massive deal. It corrected anatomists who, since 1672, mistakenly thought the entire ovarian follicle was the egg.
Oh, I see. But the real visual breakthrough came in 1876 from Oscar Hertwig. He cleverly decided to use sea urchins because their eggs are transparent.
Oh, that's smart.
Yeah, he became the first person to look through a microscope and actually watch a single sperm nucleus penetrate an egg and fuse with the egg's nucleus. He saw two distinct cells uniting.
Goodbye, homunculus.
Exactly. The preformationist idea was completely dismantled right there. And then a few decades later, around 1902, Theodore Boveri linked Hertwig's visual proof to the actual behavior of chromosomes inside those cells.
Right, the chromosome theory of heredity.
Yes, finally proving that both the maternal and paternal sides were contributing equally to the genetic blueprint.
Okay, so now that we know what fertilization actually is biologically, the union of these two incredibly specialized cells, let's look at the modern understanding of the journey itself,
Let's do it.
because it is far from the simple foot race you see in middle school health class textbooks. I mean, the numbers game is brutal. A human ejaculation contains roughly 200 to 300 million sperm.
It's an astronomical number.
But out of those hundreds of millions, only a few hundred to a few thousand ever even reach the ampulla, the widened outer third of the fallopian tube where fertilization actually happens.
Right, because the environment is extraordinarily hostile. The vast majority of those cells are neutralized by vaginal acidity, or they get physically ensnared in the complex web of cervical mucus.
Or they just get lost, right?
Yeah, they simply take a wrong turn and swim down the empty fallopian tube.
It totally shatters that myth of millions of sperm racing toward a passive egg where the fastest, strongest swimmer just wins the prize.
It's definitely not a simple race.
Most sperm are eliminated by the environment long before they even get close. But what about the survivors? Once they reach the ampulla, they just fertilize the egg, right?
What's fascinating here is that even the sperm that do make it to the egg are fundamentally incapable of fertilizing it upon arrival.
Wait, really?
Yeah. They need a biochemical upgrade called capacitation. This was discovered independently in 1951 by reproductive biologists Colin Russell Austin and Min Chu Chang.
What do you mean they aren't ready? What physically has to happen to them?
So, freshly ejaculated sperm have a membrane packed with cholesterol, which makes it very rigid. As they spend time traveling through the female reproductive tract, the local environment actively strips that cholesterol and various glycoproteins away from the sperm's membrane.
Okay, so it alters the membrane itself.
Exactly. And this does two critical things. First, it makes the membrane much more fluid and unstable. Second, this newfound fluidity allows calcium ions to flood into the sperm, which hyperactivates the tail.
Hyperactivates it.
Yeah, it goes from swimming in a straight line to this frantic, whip-like thrashing.
Wow.
The female reproductive tract actively primes the sperm, and this single discovery is why, if you are doing in vitro fertilization or IVF, you can't just mix fresh sperm and eggs in a dish.
Oh, you have to mimic that process.
Right. You have to put the sperm through a chemical incubation step first to simulate capacitation.
That is so interesting.
Gosh.
But even after they're capacitated and thrashing around, the egg is not just sitting there waiting to be conquered.
Not at all.
It is a highly active gatekeeper wrapped in a serious defensive shell called the zona pellucida.
Yes. The egg is surrounded by this thick, specialized glycoprotein coat made of proteins called ZP1, ZP2, and ZP3, and it acts as a highly specific lock.
A lock.
Yeah. For example, a human sperm cannot fertilize a hamster egg because the proteins on the sperm do not recognize the ZP proteins on the egg. It is a strict species-specific recognition system.
So how does the sperm actually get through that thick shell once it recognizes the lock?
Well, when the capacitated sperm binds to the zona pellucida, it triggers the acrosomal reaction.
The acrosomal reaction.
Right. The sperm has a small structure over its head derived from the Golgi apparatus called the acrosome. When it binds to the ZP proteins, that cap ruptures.
Like a payload.
Exactly like a payload. It unleashes enzymes like hyaluronidase and acrosin. These enzymes act like chemical drills, physically dissolving a path through the zona pellucida.
Man, that's intense.
It is. And only after it burns through that outer shell can the sperm's plasma membrane finally fuse with the egg's plasma membrane.
This completely shifts the paradigm. It's not a passive egg waiting for an active sperm. It is a highly orchestrated biochemical lock and key mechanism, and the egg is holding the lock.
Precisely.
So the lock is picked, the membranes fuse. This is where I always thought we get that literal spark of life, right? An electrical zap that shuts the door behind the winning sperm.
That is the classic fast block to polyspermy taught for decades, mostly based on Hertwig's old sea urchin models we talked about earlier.
Right, the sea urchins.
In sea urchins, the moment a sperm enters, there is an instantaneous electrical depolarization, a spark that zaps the membrane and instantly repels other sperm.
But not in humans.
No. In mammals, including humans, we don't rely on an electrical spark. We rely on a slower chemical wave.
How does a chemical create a wave?
So when the mammalian sperm fuses, it drops a very specific signaling enzyme into the egg cytoplasm called PLC-zeta. This was discovered by researchers like Jaffa and Miyazaki.
Okay, PLC-zeta.
Right. This enzyme acts like a chemical alarm clock. It cleaves molecules inside the egg that force internal storage sacs to dump massive amounts of calcium into the main body of the cell.
Oh, wow.
But it's not just one wave it pulses. The egg experiences these massive calcium oscillations for 20 to 30 minutes.
What does this pulsing wave actually do to the egg? Like, what's the purpose?
The calcium wave acts as the true catalyst for the boot-up sequence, executing 3 distinct functions simultaneously.
Okay, what's the first one?
First, it triggers the cortical or zona reaction. Thousands of tiny vesicles situated just under the egg surface are pushed outward, dumping their contents into the space around the egg.
Like a barricade.
Exactly. These enzymes physically cross-link the proteins of the zona pellucida. They take that soft glycoprotein shell and cure it into an absolute impenetrable barricade. That is the mammalion's slow block to polyspermy. The gate is permanently locked.
No more sperm getting in. Okay, what's the second function?
It wakes the egg up from a dormant state. The egg has actually been arrested in metaphase of meiosis II.
Wait, arrested? You mean the egg zone development is basically paused mid-cell division, just waiting for this calcium alarm clock to go off?
Precisely. The egg hasn't even finished preparing its own DNA yet. The calcium surge tells it to resume and complete its final meiotic cell division.
That's crazy. And the third function?
And third, the wave kickstarts the biochemical program to build the pronuclei, the physical structures that will eventually house the 2 sets of DNA.
Here's where it gets really interesting for me. The egg is basically acting like a cellular bouncer.
Yeah, that's a good way to put it.
It accepts the sperm's DNA, sure, but then it explicitly hunts down and destroys the sperm's mitochondria.
Yeah, it does.
Every biology student learns that we only inherit mitochondrial DNA from our mothers. But it's usually explained as just, a numbers game. Like the egg has 100,000 mitochondria, and the sperm only brings about 100 in its tail, so they just get diluted and lost in the mix.
Right, that's the common misconception.
But that's not true. Why go to the active effort of destroying them instead of just ignoring them?
It comes down to an active enforcement mechanism to prevent evolutionary conflict. If you have 2 different sets of mitochondrial DNA replicating inside a cell, they can compete for resources and cause severe cellular dysfunction.
Ah, so it's a safety matter.
Exactly. So, the egg takes no chances. The sperm's tiny handful of mitochondria are explicitly tagged with a molecule called ubiquitin.
Ubiquitin.
Yeah. In cellular biology, ubiquitin is the kiss of death. It's a tag that marks a structure to be shredded by the cell's waste disposal machinery. The egg specifically recognizes tags and obliterates the paternal mitochondria shortly after entry.
Which guarantees strictly maternal inheritance.
Yes, and that active destruction is why tracing human ancestry through mitochondrial Eve studies is so incredibly reliable. The paternal noise isn't just diluted, it is actively silenced by the egg.
So we have shredded the mitochondria, we fortified the walls. All that is left are the 2 sets of DNA, one from the mother, one from the father. We just mix them.
Well, not quite that simple.
Because for a long time, scientists assumed a chromosome was a chromosome. A set of 23 from a sperm and a set of 23 from an egg were functionally identical. You just needed 46 total to boot up the system.
But 1984 completely changed that assumption. 2 independent research groups, one led by Salter and McGrath and another by Serrani, conducted micromanipulation experiments on mouse zygotes.
What did they do?
They created zygotes with 2 maternal pronuclei, meaning all the DNA was from the mother. And they created zygotes with 2 paternal pronuclei, where all the DNA came from the father.
Okay.
If the DNA was truly interchangeable, those embryos should have developed normally, since they had the correct total number of 46 chromosomes.
But they didn't.
No, they didn't. They all died early or developed severely abnormally.
I get that we need one of each, but how does the cell actually know which DNA came from the father and which from the mother? Are there physical tags on the chromosomes?
There are. It is a process called genomic imprinting. Maternal and paternal genomes are not interchangeable because crucial developmental genes are epigenetically marked differently depending on their origin.
Epigenetically marked.
Yeah, think of it like a chemical stop sign specifically, a process called DNA methylation. Small chemical groups are attached to the DNA sequence, which effectively mutes certain genes.
So the mother and father have different genes muted.
Exactly. The mother's DNA has certain genes muted, and the father's DNA has different genes muted. You absolutely must have one of each complementary set for the system to boot up properly.
And we actually see what happens when this fails outside the lab, naturally, when fertilization goes wrong.
We do. In clinical conditions known as hydatiform moles, if a sperm fertilizes an egg that somehow lost its own nucleus, the sperm's chromosomes will duplicate themselves to reach 46.
So you have a full set, but they are all paternal.
Right. And because the imprinting is unbalanced, this results in a complete hydatidiform mole. It produces a disorganized tissue mass, typically overgrown placental tissue, but absolutely no embryo forms.
Or, if two sperm manage to beat the cortical wall and fertilize a single egg, you get 69 chromosomes.
Yes, this is a partial mole, and it's also non-viable. Both are powerful real-world examples of why the precise merging of one maternal and one paternal genome is so vital.
Which brings us to the ultimate culmination of this cell's journey.
The final step of this 24-hour cascade is syngamy.
Syngamy.
Right. Only after the calcium waves, the wall hardening, the meiosis completion, and the pronuclei formation do those two pronuclear membranes finally break down physically.
So they finally mix.
Yes. The maternal and paternal chromosomes intermingle in preparation for the very first mitotic cell division. Only at that exact point syngamy does a true diploid zygote exist.
Okay, we've spent the last 15 minutes deep inside a microscope, tracking calcium waves and chemical drills. But earlier we mentioned this matters in the real world.
It really does.
How do these microscopic definitions, fusion versus syngamy versus later development, actually change how medicine is practiced and regulated?
Well, how we define the culmination of this process has massive clinical and legal implications. Now, before we dive into this, we need to be very clear for you listening.
Yes, absolutely. We are neutrally reporting the biological and clinical definitions established by medical bodies and regulatory agencies found in our source material.
Right. We are looking strictly at the objective medical realities without endorsing any political side of the debates surrounding personhood or reproductive rights.
The biology is what it is, and our goal is to convey the science as it operates in medicine today. So how do clinicians define the start of a pregnancy?
The biological definition of pregnancy used by the American College of Obstetricians and Gynecologists, or ACOG, does not begin at sperm-egg fusion nor at syngamy. ACOG defines pregnancy as beginning at implantation.
Let's clarify that timeline. When you say implantation, you mean roughly 6 to 10 days after fertilization.
Correct.
When the zygote has gone through cleavage, and just to be clear, when you say cleavage, you mean those first few days where the cell just keeps dividing as it travels down the fallopian tube.
Yes, exactly that.
Yeah.
The zygote divides into 2 cells, then 4, then 8, eventually forming a hollow ball of cells called a blastocyst.
Okay, a blastocyst.
It is only when that blastocyst physically embeds itself into the uterine lining, which is implantation, that ACOG considers a pregnancy to be established.
This is the point where the body starts producing HCG, the hormone that home pregnancy tests detect.
Yes, exactly.
And that definitional choice directly dictates how medications are classified by agencies like the FDA. Take emergency contraception, like Plan B.
Right.
For years, the FDA label language stated that it worked by preventing a fertilized egg from implanting. That made a lot of people believe it was a post-fertilization drug that disrupted a zygote.
But as the science evolved, subsequent research clarified the actual mechanism. In 2013, the FDA revised Plan B's label to reflect this.
And what does it say now?
The dominant and likely sole mechanism of that drug is delaying or inhibiting ovulation entirely, meaning it acts before fertilization could ever physically occur.
It doesn't disrupt a fertilized egg. It prevents the egg from being released to meet the sperm in the first place.
Precisely.
Understanding this 24-hour timeline and the days of cellular cleavage that follow clears up so many everyday misconceptions. Like identical twins. I always assumed identical twins happened right at the moment of fertilization.
Oh, really?
Yeah. Like the sperm hits the egg and it immediately shatters into 2 identical pieces.
That is a very common misconception. It is actually the timing of the split over the next 2 weeks of early development that dictates how twins form.
So it happens later.
Much later. If the splitting happens during early cleavage, before day 4, the twins will develop with completely separate placentas and amniotic sacs.
Okay, and if it's later?
If they split between days 4 and 8, they share a placenta but have separate sacs.
And if they split even later than that?
If they split between days 8 and 12, they share a single placenta and a single sac, which is a much higher risk pregnancy. And if the split happens after day 13, the separation is often incomplete, which leads to conjoined twins.
Wow. So the later the split in this fragile timeline, the more physically intertwined the development.
Exactly.
It really highlights the sheer fragility of the entire biological project.
Yeah.
Because this cascade involves so many discrete, individually vulnerable steps. You've got capacitation, the acrosomal drill, the calcium waves, imprinting, syngamy, and then days of cleavage before implantation. It is a precarious journey.
It's incredibly precarious. Clinical embryology estimates that a substantial fraction of fertilized eggs never successfully implant, or they simply arrest during those early cellular divisions.
Fertilization is simply not the guaranteed final step that many assume it is. It is the beginning of a gauntlet.
A gauntlet is the perfect word for it.
But it's a gauntlet we've actually learned to navigate and master in the lab, which is an incredible triumph. I want you to tell the listener about how we managed to bypass these hurdles, starting back in the 60s.
Well, the history of IVF is a testament to mastering these microscopic steps. Robert Edwards and gynecologist Patrick Steptoe spent the 1960s and 70s figuring out how to mature human eggs and fertilize them outside the body.
Which was incredibly controversial at the time.
Oh, they faced massive public backlash and were repeatedly denied grants. But their persistence led to the birth of Louise Brown, the first IVF baby, in 1978.
Amazing.
Edwards eventually won the Nobel Prize for it in 2010, and IVF only works because they deciphered the biochemistry to artificially replicate capacitation and the acrosomal reaction in a lab environment.
And we didn't stop there. We eventually learned how to bypass the biological lock entirely.
Indeed, we did. In 1992, Gian Piero Palermo developed ICSI.
ICSI.
Yeah, intracytoplasmic sperm injection. This is where you physically inject a single sperm directly into the egg's cytoplasm, completely bypassing the zona pellucida lock and key system.
Incredible.
It revolutionized treatment for severe male infertility. It allowed millions of families to exist today by mastering those highly vulnerable biochemical steps.
It is breathtaking when you step back and look at it.
Yeah.
We have gone from believing there was a tiny, fully formed human curled up in a sperm cell to mapping out the calcium waves, ubiquitin tags, and methylation that orchestrate the first 24 hours of existence.
We've come a very long way.
We really have. But as we wrap up, I want to leave you, the listener, with something to ponder about the shadow side of our desire to master this process. Remember the computer boot up sequence we talked about at the very beginning?
Right.
We know all the passwords now, but we still have to enter them perfectly. And sometimes, the intense desire to control the origin of life leads science vastly astray. I want you to tell the listener about the 2005 Wang-Wusek fraud.
It remains a very dark chapter in developmental biology. This South Korean researcher published highly celebrated papers in the journal Science, claiming he had successfully created patient-specific human embryonic stem cell lines through cloning.
Cloning human cells.
Specifically, a technique called somatic cell nuclear transfer. He claimed he had bypassed the need for a sperm entirely, manually reprogramming a human egg to boot up with an adult skin cell's DNA.
And the scientific community was stunned.
Absolutely stunned. But investigations quickly revealed the data was entirely fabricated. He hadn't cloned human embryos at all.
Wow.
And worse, the human eggs he used had been obtained under incredibly ethically dubious conditions from his own junior researchers.
It set public trust in embryo research back by years. But beyond the profound ethical failures, I want you to consider the sheer hubris involved in this story. The hubris required to believe we could easily fake or casually bypass a perfectly orchestrated 24-hour biological symphony.
It's astonishing.
A sequence forged over millions of years of evolution, reliant on precise calcium waves, epigenetic imprinting, and microscopic cellular gates. The moment of conception is a myth.
It really is.
But the reality, a precarious day-long biochemical dance on the edge of a knife, is far more awe-inspiring and far more difficult to control than we ever imagined. Let that linger next time you think about where we all came from.